my name is Siting and I'll be presenting alongside Herman, Junius, Zipin, Xavier and Nigel. Today we'll be talking about biofuels. Our agenda today will be introducing you to biofuels, sharing our justification, having a literacy review, explaining our biofuel experiment, showing our timeline, and last but not least, we will have a summary to sum up everything we mentioned today. So first, I'll be introducing you to biofuels. You may be wondering, what are biofuels? Biofuels are fuels derived from living matter. Although we have fossil fuels, it will eventually be used up. Thus, by using biofuels, fossil fuels can be sustained indefinitely. But why did we choose biofuels? We came across Singapore's International Water Week, and we stumbled upon exhibitions that showcased the uses of biofuels. This sparked our interest, and we decided to explore further and use this topic for our project. Now, I'm going to share some of the exhibitions that we saw. Number 1 shows how food waste is turned into substrate for microalgae production that can be used for food supplements and/or biofuels. Number 2 shows how the use of AI can sort circuit board components at high efficiency to recycle or recover resources. Number 3 shows how plastic waste can be turned into permeable road so that water can seep through and into the ground when raining to prevent flooding. Last but not least, number 4 shows how utilizing microorganisms can turn industrial waste into fish food and/or fertilizer. Now I'll be handing over to Herman, who will be sharing the experiments that focuses on food wastage in Singapore.
Thank you, Siting, for the introduction. We'll now be turning our focus on two prospective projects that lead us to leverage biofuel. One is a bioreactor that utilizes industrial food waste to feed purple photogenic bacteria, which are then collected as biomass for fish food and fertilizer. The second involves using microwave to break down food waste into biomolecules to feed and cultivate algae for fuel and food production. Both purposes involve recycling food waste into useful product, but they may not be economically viable. That's why, moving on to our problem statement, we ask, how might we design a low-cost, environmental-friendly way to encourage corporations to recycle their food waste to reduce carbon emission and gain benefit while doing so? We first have to understand the relevancy of food waste in Singapore. Did you know that in 2023, 11% of the total waste generated came from food waste? To add to the problem, our recycling rate has declined to 52% over the years. If the amount of food waste generated keeps getting higher and the amount recycled keeps getting lower, we'll be seeing more waste sent to our landfill, which is what we don't want. Furthermore, food waste also impacts Singapore economically and environmentally. A staggering $342 million were lost to food waste alone. To add fuel to the fire, for every 1 kg of food waste, it contributes 2.5 kg of carbon emission to the environment, which is why it's so hot in Singapore nowadays, don't you all agree? This ties us to the next slides. Here, we discuss the impact of global warming brought by the enhanced greenhouse effect, which is affected by the increase in carbon emission. So, what are the impacts of global warming? As you all know, one of the impacts is sea level rise. As you can see, in the recent news, the residents of Tulava are already preparing to migrate due to sea level rise, which shows that global warming is already here with us. Besides sea level rise, we also have to be more cautious of increased insect-borne diseases, as cases of dengue are seen on the rise recently, with 7 reported deaths as of 2024. Singapore will also need to prepare for a hotter, wetter future, as weather phenomena are intensified due to global warming. Last but not least, global warming also threatens Singapore's food security. With us importing over 90% of our food, a global food shortage would heavily impact us. Moving on to the next slide, we discuss the current context. Singapore is already preparing for a future through the SG Green Plan 2030, which charts Singapore's ambitious and concrete target over the next 10 years. One of the aim is to reduce waste sent to landfill by 30%. This is significant because the Semakau landfill will be full in the next coming decade. To tackle this issue, our team wants to turn food waste into biofuel, which is the third option in the waste hierarchy. Now, I'll hand over to Zipin to tell us more about biofuel.
So let's understand what exactly are biofuels. Biofuels are fuels that are derived from renewable biological resources or biomass. They are produced from organic matter such as plants, crops, algae, and animal waste. And they are also considered as a form of renewable energy, as the biomass used for their production can be replenished through natural processes over time. Using biofuels also bring about several benefits. Not only does using it reduces greenhouse gas emission as compared to using fossil fuels, it also does not contribute to air pollution. Moreover, it's renewable and sustainable while also being cost-effective at the same time. Some examples of biofuels are bioethanol, biodiesel, biogas, bioethers, biojet fuels, and synthetic biofuels. So, the process that we have decided to conduct an experiment on is methanogenesis. Methanogenesis is the production of methane caused by bacteria or other living organisms. It is carried out exclusively by a group of anaerobic archaea, commonly known as methanogens, which are distinct from bacteria but are closely associated with anaerobic bacteria. The process of methanogenesis involves a series of biochemical reactions that convert simple carbon compounds such as carbon dioxide, acetate, and methanol into methane, which can then be used to generate energy. Some of the biomolecules that are involved in our system process are carbon dioxide, hydrogen gas, acetate, methane, and hydrogen sulfide.
Methanogenesis is one method for recycling food waste, but there are two additional techniques: harvesting black soldier fly larvae and cultivating purple phototrophic bacteria. Each of these systems utilizes different types of food waste and operates with distinct processes and outputs. Methanogenesis involves a batch process where organic food waste is placed into a digester. Over time, microorganisms break down the waste anaerobically, producing methane, which can be captured and used as a renewable energy source. The process requires careful management of temperature, pH, and other conditions to optimize methane production. Harvesting black soldier fly larvae operates as a continuous process. Vegetable scraps, fly, fruit peels, and other types of food waste are fed continuously to the larvae. As the larvae consume the waste, they grow and are harvested periodically. The output of this process is frass, a nutrient-rich byproduct that can be used as fertilizer for local vegetable farms or as feed for the tilapia fish, which provide an additional food source. Cultivating purple phototrophic bacteria also involves a batch process. Industrial food waste is used as a growth medium for these bacteria in specially designed photobioreactors. The bacteria convert the waste into valuable biomass, which is harvested and can be used as fertilizer in agriculture or as feed for fish. This process requires control over light conditions and other environmental factors to ensure optimal bacterial growth and biomass production. In summary, while methanogenesis and purple phototrophic bacteria cultivation rely on batch processes with distinct waste inputs and outputs, black soldier fly larvae harvesting is a continuous process that integrates waste cycling with agricultural and aquaculture applications.
Materials used for our experiment consist of 5 plastic bottles that were taken from waste bins all around Ngee Polytechnic and were washed thoroughly before being used for our experiment. Pond water that was collected from Commonwealth Secondary School that contained anaerobic bacteria as well as some of the sediment in the pond. Food scraps such as radish and carrot peels that were left over from cooking. Rubber balloons that were wrapped around the bottle to collect any of the gases produced by the bacteria. And finally, tape that was wrapped around the bottleneck to ensure that there would be no gas leaks. Firstly, pond water and sediment were scooped into the bottles. Secondly, an equal amount of vegetable peels were deposited into each bottle. Thirdly, we wrapped the rubber balloons around the bottleneck to collect any of the gases that will be produced. Fourthly, we observed day by day the amount of gas produced by the anaerobic bacteria and correlated it to the size of the balloon. And lastly, we noted down any fluctuations in the size of the balloon and checked for any damages to the experiment before we noted down the size change. So here we have the system process of our experiment. Food waste such as discarded vegetable peels go in, and the vessel is sealed after that. And the methanogenesis process will turn the food waste into methane that takes from 2 to 3 weeks. And when the vessel is finally opened, we can collect the methane inside. Here, analysis of our experiment. Phase 1, aerobic decomposition, which takes place from July 1st to July 2nd. On the first day, when the food scraps were added to the bottles containing the pond water, balloons were then attached to capture any gases produced. Initially, there were no noticeable changes to the bottles. However, by July 2nd, the balloons began to deplete. This suggests that aerobic bacteria in the bottle were consuming the available oxygen in the sealed environment. In Phase 2, the transition to anaerobic conditions takes place from July 3rd to July 4th. By July 3rd, the balloon stopped deflating, which indicates that the oxygen inside has been depleted, and the appearance of gas bubbles on the vegetable peels suggests the onset of anaerobic decomposition. On July 4th, the balloon started to inflate slowly, which confirmed the production of gases such as carbon dioxide by the anaerobic bacteria. During Phase 3, which was peak anaerobic activity, lasted from July 5th to July 10th. This phase showed significant anaerobic activity, such as 3 out of the 5 balloons inflating enough to be able to stand on their own, more gas bubbles being observed on the surface of the vegetable peels. A putrid smell was noted when one of the balloons broke and had to be replaced, which indicates the production of hydrogen sulfide, a common byproduct of anaerobic decomposition. Carbon dioxide was also likely produced during this phase. And by July 10th, the balloon started deflating slowly, which suggests that the anaerobic bacteria were consuming some of the produced gases. Phase 4, which is depletion and transition, lasted from July 15th to July 17th. The vacuum effect that was observed on July 15th indicates that the gases were being consumed at a faster rate than they were being produced. This could be due to the depletion of easily digestible organic matter, such as the vegetable peels, or a shift in the microbial community. By July 17th, the vacuum effect stopped and gas bubbles could be seen rising from the sediment layer when the bottles were slowly rotated. This suggests a new phase of decomposition, which could possibly involve different types of substrate or bacterial populations. And finally, the last phase, which is Phase 5, methane production, which was on July 19th. The rapid inflation of the balloons, and one of them standing up due to the gas buildup, indicates that the production of methane was underway. Methane is typically produced in the later stages of anaerobic decomposition by methanogenic archaea. The conclusion that we drew from this experiment was that it demonstrates the various stages of anaerobic decomposition, the initial aerobic phase, the transitioning to anaerobic conditions, the peak anaerobic activity with production of CO2 and H2S, a period of gas consumption or reduced production, and finally the last stage, final stage of methane production. How we can improve our experiments? Firstly, we could include a control in our experiments to ensure that the gas produced is from the microbes breaking down the food waste and not from any other factors. Secondly, a quantitative way to measure the gas produced over time, such as a gas collection tube, will also be very helpful. Thirdly, using what we learned in IoT to track the rate of change of methane concentration over time. And lastly, the inclusion of a more dependent variable such as carbs or proteins and lipids and dietary fiber, as well as a higher temperature or room temperature to see whether that affects the production of methane.
Biofuels are a renewable source of energy produced through a process called methanogenesis. In this process, methane is actually generated from food waste and organic matter. This methane is then converted into energy through combustion, producing a substantial amount of heat, making it efficient and effective heat source. Some environmental benefits that actually brings us is actually energy production through this process, as it actually offers significant environmental benefits. It greatly reduces carbon emissions, thereby helping to mitigate global warming and greenhouse effect. In contrast to fossil fuels, which emit higher levels of harmful greenhouse gases like nitrogen oxide, biofuels actually provide a cleaner and more sustainable alternative. So not only that, sustainability wise, one of the key advantages of biofuels in their sustainability is that food scraps and dead organic matter can be easily replenished, making biofuels a renewable resource. This contrasts sharply with fossil fuels, which are finite and contribute to environmental degradation. By using biofuels, we can actually reduce our reliance on fossil fuels and promote a more sustainable energy future. So this project actually aims to explore more innovative ways into biofuels, and we have several key objectives in this project, which are to clear up landfills, which is particularly important in land-scarce countries like Singapore, promote environmental sustainability, provide cost-effective solutions to combat climate change, which we have actually narrowed it down to biofuels. After thorough exploration and analysis, biofuels are actually the most optimal solution to achieve these objectives, and by leveraging methanogenesis, we actually can transform food waste and organic matter into valuable energy sources. Not only that, we can actually reduce landfill waste and contribute to a cleaner environment. So actually, in conclusion, biofuels actually represent a promising renewable energy source that offers numerous environmental sustainability benefits, and this project actually aims to harness this potential to create a more sustainable future for Singapore. Methanogenesis is actually a critical process where methanogenic archaea degrades organic compounds under anaerobic conditions to produce methane. Actually, this primarily occurs when aerobic bacteria depletes oxygen. Hence, recognizing this process is actually essential for applying our findings into real-world challenges such as reducing food waste and landfill accumulation. How would we actually solve this problem? So first off, waste management. Methanogenic technology actually offers an efficient solution for converting organic waste into biogas, a renewable energy source, and this reduces our dependency on fossil fuels and decreases the volume of waste requiring landfill space. So in a land-scarce country like Singapore, this is particularly beneficial. By minimizing landfill waste, we can actually mitigate the environmental and societal impacts associated with waste disposal. And not only that, this also provides a sustainable agriculture for Singapore. How is this so? The byproducts of methanogenesis can actually be repurposed as nutrient-rich biofertilizers. These biofertilizers actually enrich the soil with essential nutrients, supporting the sustainable agricultural process. This is especially advantageous for urban farming initiatives in densely populated areas. Utilizing biofertilizers can enhance crop yields and promote a more sustainable food production system. This is not only sustainable in agricultural terms, it is also resource efficient. So we can actually transform organic waste into valuable resources like biogas and biofertilizers, which can exemplify into principles of a circular economy. This approach not only maximizes the resource efficiency by ensuring that waste products are not discarded, but also converted into useful outputs. This not only minimizes the environmental impact, but also promotes the sustainable use of resources, contributing to long-term environmental conservation. Lastly, through this initiative, that we've also found that innovation is actually significant to our project, as investing in methanogenic technology actually drives research and development, positioning Singapore as a leader in sustainable tech. This actually fosters innovation and creates new opportunities for economic growth and development. By leading in this field, Singapore actually can set an example for other nations, demonstrating how advanced technology and sustainable practices can work together to address global environmental challenges. In conclusion, the significance of our project actually lies in its multi-faceted benefits. By leveraging methanogenesis, we can actually effectively manage waste, promote sustainable agriculture, and also enhance resource efficiency and drive innovation. This holistic approach not only addresses immediate environmental concerns, but also paves the way for a more sustainable and prosperous future.